So now I'd like to talk to you about what happens when you have these monomers and they join together to form polymers, when the nucleotides join together to form nucleic acids. Now recall that it starts out as with the nucleic acids, you have the nucleotides joined together by that phosphodiester bond, and it runs from 5' to 3', and then it's going to be paired up with another strand, a different molecule, and this strand, these strands are going to interact. It's going to be interactions between them, and there's a few different interactions that occur. For instance, one of the key ones is the hydrophobic effect, and so they call that stacking, where they kind of come together to repel water, and whereas the phosphates on the outside is very hydrophilic, and on the inside these bases are somewhat hydrophilic but also have hydrophobic elements as well, and so we're going to kind of stack together a little bit to exclude water. But also one of the major ones are hydrogen bonds. And the hydrogen bonds in particular are really important for making sure that they, the bases line up correctly and make the correct pairings. And this pairing gives us data redundancy. Because if, for instance, if you've got these pairs here, if something were to happen to one of them, well, since we already have this one, we know that, let's say this one is an A, we know that this one must be a T, which means that the biology, the cell can read, say, hey, this is an A, this one's missing, but it must be a T, so I can put in a fresh T if something were to happen. So it adds a data redundancy, which is good, but we need to make sure that they always line up correctly. And what's responsible for this are hydrogen bonds. Now, here on the left, we've got adenine and guanine, and notice that these are purines. This is the more complicated structure with the simpler word. So these are the purine, there's the 6-membered ring and the 5-membered ring attached to it. And on this side, we've got the pyrimidines. And this is the simpler structure that resembles pyrimidine, but it's got the modifications to it. So a purine always pairs up with a pyrimidine. You see purine, pyrimidine, purine, pyrimidine. This always happens. But the reason why they always pair up correctly is because the correct hydrogen bonds. Let's start out with adenine. If we look at the top, we see that we have this amine group, and this amine group has got a this hydrogen here. This hydrogen is acting as a hydrogen bond donor. And what is orientated just right, the correct distance, around about 3 angstroms, is perfect for the formation of a hydrogen bond. Also the bond angle. Also, this is a donor pairing up with an acceptor. If it was a donor and a donor, they wouldn't pair up, but it's a donor with an acceptor. That's just right, because this carbonyl was acting as a hydrogen bond acceptor, the lone pair of electrons on the oxygen. On the other hand, down here, we've got this nitrogen here of the heterocycle, and this is going to act as a hydrogen bond acceptor, being paired up with this hydrogen attached to the nitrogen of the ring, which is going to be a donor. So the acceptors with a donor, the donors with an acceptor. The distance is just right. The angle is just right. The presence of these 2 hydrogen bonds, the conditions are just perfect for these to pair up. On the other hand, if we look at the G and the C, guanine, cytosine, we notice that there's now 3 hydrogen bonds instead of 2. So it's 3 with GC, 2 with AT. But notice now we have this combination of acceptor, donor, 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 acceptor, acceptor. Whereas here it's donor, acceptor. So it's the other way around, acceptor, donor, donor, acceptor. So, you know, it's a different combination. You have a different number of hydrogen bonds, but you also have whether or not it's a donor or acceptor combination being the right or the wrong way around. You have the distance being just right and the angle being just right. And if you were to have a different, you wouldn't be able to fit a different base in here. You wouldn't be able to put thymine in here, because not only would it be mismatching 3 and 2, but also you'd have an acceptor pairing with an acceptor. It just wouldn't work. So you've got to get this all right. Or I mean, if you were to flip it upside down, it would also you'd have this all pointing up here. It wouldn't work. So the bases pair up because the combination of the hydrogen bonds is just right. And you can see that here. If we this is just a bigger illustration of what we're looking at. And we can see that this will go all the way down the strand from 5' down to 3', all the way down, and the C will pair up with the G with 3, and the A and the T with 2 hydrogen bonds. Notice as well what we can see here is that if this is the sugar, and we've got 1, 2, 3, 4, 5, and there's the 5' phosphate at the top, but notice that 3' is attached to the phosphate of the one next in sequence, and then 1, 2, 3, 4, 5'. So it's connected here. So the 5' is always at the top because that's the phosphate which is unconnected, because that's just the first one, so it doesn't have anything. And then the 3' is at the bottom, and it connects to the next phosphate, which is on the next base attached to the 5' position, until you get all the way down, and eventually you have a 1, 2, 3', which doesn't have anything attached to it. So that's the 3' end. And this runs anti-parallel to the other strand, which runs 5' up to 3'. So it always runs 5 to 3, 5 to 3, anti-parallel to each other. And this kind of describes here. 5 to 3, 5 to 3, running anti-parallel to each other, complementary. C with G 3 times, A to T 2 times, with the correct combination of hydrogen bonds. Now, once you have these strands of DNA, this double-stranded, which are interacting with each other. It's similar with the proteins, where you had a polypeptide chain, but then that polypeptide chain can form all kinds of like local folding. But then that local folding will come together to form a whole structure of a protein, which may be, forming like a pocket for a substrate to fit into, or maybe to form some kind of strand which has like structural integrity. In the same way, DNA also will fold, and that's how you can fit all your DNA in your cell. Your DNA has got so many genes in it, so many bases, so many genes. It's so long. I'm not sure exactly, but I think I read something like if you took out all of the DNA, or just from one single cell, just took the DNA out and just stretched it out longwise, it would reach the moon. I can't remember what it is, but it's some sort of absurd long distance, right, the DNA. And you've got that in every single cell of your body. There's every single cell in your body has got your entire genome in it. So how can you have that? How can you have such a long thing and such a small thing so many times? Your DNA isn't shared between your body. Every single cell has its own copy. How can you fit it in? Well, it folds and gets very, very tight and compact. So that in the same way as you had the primary, secondary, and tertiary structure of the nucleotides, you've also of the polypeptides and amino acids to proteins. You've got the same thing with the nucleic acids. You have the primary structure, which is just the nucleotide sequence running 5 to 3. And that was just to say A, C, A, G, C, A, C, G, A, T, something like that. That's just the primary sequence. Then you have the secondary sequence. Now this is, again, the same kind of thing as the secondary sequence in the nucleotide, I keep saying that, in the polypeptides, where you have local folding, and that's actually the helix. The helix is the secondary structure. And the way that, you have the double-stranded, but then the double-stranded twists in order to form a double helix with a major and a minor groove, which I think we'll mention a little bit later. There's the double helix. The tertiary structure is then when it winds even further in order to make the chromosomes. And what happens is that the DNA will wind around proteins called histones, which are just kind of like little balls. Like if you had a elastic band ball and you were to wrap it around the ping pong ball in order to get that elastic band down, the histones would kind of be like the ping pong ball. You can kind of think of it as being your elastic band chain. Maybe you'd be able to stretch it around the whole of campus. But if you were to wrap it around the ping pong ball, you'd probably be able to get it into something maybe the size of a football or something like that. So that huge long thing, you'll be able to wind and make it really small. You do it by wrapping it around, for instance, the histones, which are proteins. Now, DNA, remember, has got a negative charge, deoxyribonucleic acid. That phosphate gives it a negative charge. Histones are proteins that have a lot of positive charge. Histones have a lot of lysine, which has a positive charge. That's NH3. I butchered it a little bit. NH3. The R group is NH3 of lysine. It's got a lot of lysines on it. And so the negative charge of DNA will attract to the positive charge of the amine group of the lysine, and that'll help it wind around, and it will wind more and more and more, tighter and tighter and tighter, put lots of them together until you get your chromosomes. And every one of your chromosomes is just tightly wound DNA that's just been wound around a bunch of histones. And actually, a small side thing is that that lysine, you may recall from a previous lecture on amino acids, lysine can be acetylated, and when it's acetylated, it loses its charge. And so histone acetylation is what causes DNA to unwind for when your genes need your machinery, cell machinery, kind of polymerases and transcription enzymes. They need to read. Well, they can't read if it's all tightly wound, so it needs to unwind slightly. And to unwind it slightly, histones get acetylated, so they lose their positive charge, so the DNA doesn't attract as much, and so it relaxes and unwinds slightly. But that's a little bit unnecessary, but I thought I would mention it. Now, this is what DNA actually looks like. This is a photo of DNA. This is actually one of the earliest photos of DNA. This is what really kind of, you may have seen this photo before. This is what gave the hint of what DNA looks like. And it probably looks like nothing to you. And the first time I looked at it, it looked like nothing. It takes a while to kind of get your head around what you're looking at here. This is a picture that was taken by a lady, Rosalind Franklin, who joint won the Nobel Prize, Watson and Crick. She doesn't get nearly as much credit as she should have done with Watson and Crick, who kind of proposed the structure. But they were able to. She took this photo. She was an X-ray crystallographer. She took this photo of DNA, and Watson and Crick were able to look at this photo and figure out what the structure of DNA looks like. They knew it was made out of nucleotides. They knew that it arranged, and those nucleotides polymerize, and then they form a structure. But they didn't know what that structure looked like. They didn't know what the double helix looked like. They were able to figure out from this what the double helix looks like. And it took me a while to be able to see it. But what you're looking at is, if you were to think of DNA, and you think, I've not done that. Let's get it to better one. DNA, and here's the double helix. What you're looking at here, this cross here, is, for instance, this here. It's this here, or this here. That's what this is. It's just a section. There's just a cross section of the DNA like that, and they were able to figure it out. And these diamonds, they were able to figure out that's the phosphate bases, and the fact that they could see them told them that the phosphate, not the phosphate bases, and phosphate backbone. The fact they were able to see this indicated to them that the phosphate must be on the outside, because they weren't sure if it's going to be outside or the inside. So this indicated that the phosphate is on the outside, because the phosphate's forming this diamond, and the fact that you can see it must mean it's on the outside. And the fact that it's like this cross indicates that it must wind as a winding helix, where you have this perhaps like ladder, but the ladder has twisted. You have this twist in the ladder. If the X-ray would have looked like that, or I guess if it would have looked like this, then they might have considered that it looks like a ladder. But the fact that it forms a cross indicates that the ladder is twisted. And also the fact that the ladder, the angle is different. The size of these triangles is different. Indicates that it's not symmetrical either. You have a major and a minor groove. So Franklin and Wilkins were originally working on this kind of work, and they figured out that the cross means that it's a helix, And then Watson and Crick were able to take this and work out that it was a type of hydrogen bonding with the bases pairing up. And the way that the pattern forms means that it's an alternating pattern of these major and the minor grooves, which means that double helix fits all of this information together, and they were able to conclude, they were able to put this together and their stuff together and conclude that it forms that double helix. And actually, I stand corrected. It seems that Watson, Crick, and Wilkins, who was another tech working with Franklin, they shared the Nobel Prize. I thought that Franklin did as well, but maybe not. My bad. Franklin, Watson and Crick was still alive a couple of years ago. I don't know if they still are, but Franklin died a long time ago. She actually sadly died of ovarian cancer, most likely due to all of the radioactive reagents that she was working with when she was doing this type of research. But essentially, these guys, these folks were all able to put together their knowledge in order to conclude the structure of DNA. And this is what this is kind of what it looks like. So this is 3 different illustrations. There's a cartoon, the full atomic, where you can see all of the atoms, and then with the atoms being arranged in a space filling kind of conformation. You can see that this is it's not just a helix, right? It's a double helix, but also it's asymmetrical. You can see here that this triangle is bigger than this triangle. This triangle is bigger than this triangle. That is a major groove and a minor groove. That was some of the information that Watson and Crick were able to work out from the way that this structure looked here. They're able to figure out every term is about 10.5 base pairs, and about a full term is about 36 Armstrongs. Now notice here it says bDNA. There's other structures of DNA as well. The classic one that you guys will be familiar with is bDNA, and that's just when DNA is solubilized in water. There's also other forms of DNA, the A form and the Z form. This isn't super important, or it is super important, but this isn't so important for the biochemistry, undergraduate biochemistry course. But I just thought I would point out that the regular structure of DNA that you guys are familiar with would be is actually just known as the B form, and it looks like this with the double helix and the bases on the inside. If you were to take DNA and dry it out, it actually forms a slightly different structure, which is known as the A form. And there's this other form as well that's been known as a Z form. And this is actually a left-handed instead of a right-handed helix. And we don't really know what this does, but it seems that it's important for gene regulation, where certain sections of DNA will just winds the wrong way, and it makes it harder to read, and thus it's their way of switching off genes. We don't really know an awful lot about it. But B-form DNA is the main one, and I just wanted to make you aware that there's other types of DNA, and we may or may not know what they do. Now, how does this all keep together? You know, we've got these huge, long structures, and they stick together, and they stick together very well. Your DNA doesn't really spontaneously unwind an awful lot. So there's a few ways that DNA keeps together. The hydrogen bonding isn't actually the main thing between the bases. The hydrogen bonding between the bases is not really the main thing. That helps to get the complementation just right, but there's other things as well. For instance, the phosphates, remember the phosphates are on the outside. Let's go back a little bit. So the phosphates are all on the outside of the strand. Here's the phosphates. Phosphate, base, sugar, base, phosphate, sugar, base, phosphate, sugar, base. The phosphates are all on the outside. There's the phosphate again in blue, and the sugar is that yellow, and the base is that yellow. The phosphates are on the outside, but the phosphates are negatively charged, and that negative charge is going to repel. So in cells, you have to have a counter ion, and that is magnesium. And the magnesium counter ion helps to keep the DNA backbone together by dispelling the negative, repelling, and negative charges of the backbone. And I remember the other one I said as well is this base stacking. It's kind of the hydrophobic effect, where the bases, notice they overlap. And where they overlap, you have a combination of the hydrophobic effect, excluding out water, but also a stacking of pi electrons that help to stabilize things, and actually has a bigger effect than the hydrogen bonding. The hydrogen bonding helps get the complementation correct, but the base stacking helps to keep it together, and the negative charge kind of counter, the magnesium ion acting as a counter ion to the negative charge of the phosphates, that also helps to keep things together. And I guess it's a shield, the negative charge of the phosphate backbones from each other. Just a single slide really on RNA. RNA, just like a DNA, except you have uracil instead of thymine, and the sugar is ribose instead of deoxyribose. So the sugar at the 1,2 prime is 2 hydrogens instead of an OH and a hydrogen. Being 1 hydrogen, not an OH, makes it deoxy. And this is ribose, so it's deoxyribose. Just that when we have kind of like the trinity, we have DNA to RNA, mRNA, I should say, really, to protein. And the DNA is the gene sequence. The RNA is then taking that gene sequence and making a copy of it. It's transcribed, and it's translated to protein that makes the protein. This is like the recipe book. This is like taking the page in the recipe book, and this is like making the cake. And but of course, the thing is that once you have the recipe book and once you have the recipe out, you can make that cake multiple times. So the mRNA can be read multiple times to make the protein multiple times. Or sometimes a recipe, the page might have multiple recipes on it. So one mRNA can sometimes code for more than one protein, because on it, it may be that one section that's transcribed into an mRNA by a transcriptase enzyme to the RNA or messenger RNA, may be transcribed, but then that may contain 2 genes or more, and then that may be, that's called polycistronic, and then that may be turned into multiple proteins. There's a lot of regulation there. There's also tRNA, which is transfer RNA. That is important. That's kind of in the, that helps with the codons of keeping translating the mRNA into the protein, is the tRNA. And tRNA is just actually just RNA that's folded and behaves like a protein. Kind of cool. So you're used to thinking of mRNA as behaving as a, it's just like the recipe in between the DNA and the protein. It's that message which is translated into the protein. And you'll think of it as just being like a messenger, a carrier. But tRNA and rRNA, ribosomal RNA, are actually RNAs that fold and also have a shape that behaves like a protein. It's like a tool. It folds to behave like a tool. So messenger RNA acts as a message, rRNA and tRNA fold to behave like tools, kind of like a protein. I thought that's kind of cool. But I don't want to talk too much about RNA because I didn't want to get too bogged down. Now, the next thing I'd like to talk about, and this is kind of the penultimate topic, is really about DNA denaturation. That is where you have the 2 strands running 5' to 3', 5' to 3', and you have the interaction between them. But if you were to apply, for instance, heat, you can get them to break apart. Because, of course, think about water. What happens? Water is held together by hydrogen bonds. If you apply heat, then those hydrogen bonds break, and the water, rather than being all kind of stuck together, breaks apart, turns into a gas, turns into steam. In the same way, you can do the same thing where you apply heat to a double-stranded DNA, and the 2 strands will break apart. So if you, in the DNA denaturation, that's just strictly refers to when you have double-stranded DNA and you break, or really any kind of double-stranded nucleic acid, but particular DNA, because DNA is double-stranded more often than RNA, which is usually single-stranded, but when it folds, it can form double-stranded regions. Anyway, the DNA, you're just separating out the two strands. So the covalent bond between that phosphoester bond between bases, that stays intact. The hydrogen bonds are broken, so the two strands separate, and the base stacking is lost. So remember that major effect that holds them together, the base stacking I brought up here. Ooh. Here, the base stacking, where they overlap, that is lost. And so because that's lost, it actually means that you have more bases exposed, and so the UV absorbance will increase. Because remember, way back, I talked about how the base absorbs UV radiation, and that's how we measure DNA concentration in the lab, things like that. Anyway, so really, you can do something to break apart the strands, but the code is still remains intact. So the denaturation can be induced by a range of things. Heat is a classic one, but also a change in pH, and also as well salt as well can mess around with it. But of course, the thing is that these are the weak intermolecular forces of hydrogen bonding and the hydrophobic effect. So these are non-covalent forces that were holding it together, and so they can be broken, but also they can come back together again. And so if you bring back down the temperature, these will join back up again. They'll join back up again to form the double-stranded again, and that is called annealing. So you can go through a cycling of, let's say, these are bases together, and you can break apart, which is denaturing. They can come back together, which is annealing. Now, this is exploited for different things such as PCR, which we'll get onto in a moment. But it's just another illustration. You have your double-stranded DNA. In blue is the phosphate backbone, and the bases interacting between it. But if you were to apply heat, you can break it apart. And then if you continue to apply that, at first it'll just partly break. If you continue to apply it, then it will completely break apart. But if you were to reduce the heat, then it will come back together in bits and then in whole. And the thing that really controls it coming apart together or in bits is to do with the, here, this is a strand of DNA. The thing to do with it coming, and you can see that here's the strand, and this is it kind of as the double-stranded. But notice in regions, it's become single-stranded. Different regions of the molecule are going to have what we call different melting temperatures. Now, the temperature at which the DNA breaks apart, that is known as the melting temperature. So the heat required to make the two strands break apart from the interaction together, break apart into the two single strands, the heat required, that's called the melting temperature. The different sections of the DNA are going to have different melting temperatures. Now, we can actually measure this using spectrophotometry. And so, for instance, what you could do is, remember I said that the absorbance changes as the DNA melts. So what you could do is that you could have denaturation on the y-axis, where you're saying that the UV increases when it melts and breaks apart. And so you can so you could have it as a percentage of denaturation, but this is actually going to be really absorbance. And then you can have the temperature increasing. And at normal temperature, you can just call it 0. We just set it to 0, whatever the absorbance is. And then as you increase the temperature, eventually you'll get to a point where the DNA starts to break apart. And those two strands start to break apart. And so the absorbance will increase. And you can measure that increase in absorbance. And if you have a whole strand of DNA, you could the temperature required for it to melt half of the strand of DNA is called the TM. And so, like I said, sections of it will melt. It'll melt bit by bit. sections of it will break apart a bit by bit, and once you have half of them broke apart, as the temperature required to hit that is known as TM. And two different strands can have two different TMs because they have can have more or less regions that have a higher or lower melting point. And this is, like I said, this is utilized for PCR. This is what I've already mentioned, the midpoint of the melting. This is really what it depends on, what causes that. So what causes this difference in TM? Now recall with the A and T, C and G pairing, recall the hydrogen bonds. A and T, 2 hydrogen bonds. C and G, 3 hydrogen bonds. So regions of the DNA which are melted at a lower temperature, they are AT rich. Regions which are still double-stranded, rather haven't melted into 2 single strands, they're going to be GC rich. So as you increase the amount of GC regions, the DNA requires more heat in order for those regions to melt, and overall the TM will increase. Also, a longer strand of DNA is going to require more heat in order to get it to melt. So a longer DNA has got a higher TM, a higher melting point. And also the, like I said, the pH and ionic strength, so salt, is also going to affect the TM as well. So this comes in important, for instance, when we're doing PCR. And if we've got a gene that we want to amplify by PCR, we've got to make sure that we apply enough heat in order to get it to separate, but not so much heat that it causes it won't the PCR enzyme won't work properly. So we need to get a right balance of the TM in the region that we want to amplify, and this is going to be affected by the pH and the ionic strength, because when you do PCR, you do it in a buffer, and the buffer is going to balance pH and ionic strength. So you want to have this kind of standardization between them. That's going to be important. If you're amplifying something shorter or longer, that's going to affect the TM as well. So really, overall thing to take away from this is that the double-stranded DNA is held together in multiple ways by weak intermolecular forces. If you apply enough heat, you can break it apart into 2 single-stranded. The temperature required for a length of DNA to get 50%, this breaking apart is called melting. It happens unevenly. Some parts will do it more than others, depending on the bases, because the bases will have different attractions for each other and require more energy and heat to break them apart. And the the heat required to break apart 50% of the DNA strand is known as its TM, and it's affected by GC, length, and ionic strength and pH. For instance, here, this graph here, as the GC increases, so does the melting point. And like I said, this is what it looks like. Double-stranded regions, when you apply heat, are probably GC rich. Bits that have started to break apart are probably AT rich. Now, detection. Okay, so if you have a kind of building on this a little bit, is the idea that if you have a this is really talking about if we want to make artificial nucleic acids and synthesize them and use them as tools for doing things. So what we could do, for instance, is that if we have a strand of DNA, double-stranded DNA, we can melt it into the 2 single strands, and then we can incubate our own single strand, which will be complementary to a certain region. And that when it becomes complementary for a certain region, we can use that to selectively bind to one part of the DNA. And then once we have that selectively bound to one part of the DNA, we could use that, for instance, if this has a tag on it, maybe a radioactive tag or a fluorescent tag, we could use it to detect whether or not that thing is present. So if you want to know whether or not a gene is present, you can break apart the DNA, melt it, put in a complementary strands that you've synthesized that's got a tag on it, lower the temperature a little bit, they will anneal, come together. It's called hybridization. And then if it sticks, then it will fluoresce or have a radioactive signal. If it doesn't stick, then it'll just get washed out and you get no signal. Or alternatively, you can use it to selectively amplify this part and do PCR on this part and make lots and lots of copies of this part, which is how PCR works. Or you can use it for mutagenesis, which is a special type of PCR. You can use it for qPCR, which is a special type of measuring RNA levels. This is, again, using artificial probes that you've synthesized that are complementary to things that you're interested in, and using that to try and glean some data from it. Also, interestingly, is that sometimes your body has its own complementary regions of DNA and RNA that it synthesizes in order to control gene regulation. For instance, mRNA is normally single-stranded, and the mRNA gets translated into a protein. But maybe it gets translated from the DNA to the transcribed from the DNA to the mRNA, but maybe you don't want to do anything with it. Maybe you don't want to turn it into a protein. Now, there's various situations where this might happen. For instance, let's say that the mRNA is the DNA is a viral DNA that's been inserted into your cells, and then your cell is then forced to make the mRNA and then forced to make the viral protein. But your body wants to fight back. It can try and cut out the DNA, but this can be difficult. Another thing it can do is to fight against the mRNA, and your body can synthesize a complementary mRNA. An mRNA is normally single-stranded. You can synthesize a complementary mRNA that will then bind, and then your body will then say, hey, there's a double-stranded mRNA. I don't like this. I'm going to chop it up. And then you don't get the protein. Or sometimes your body constantly makes the mRNA, so you don't have to switch the gene on or off. You just constantly make out of the mRNA, and then it's already sitting there ready to go, and it's going to get translated into a protein. But something jumps in at the translation step rather than the transcription step, because it's faster, because the mRNA is already there. So you can have a faster response to a stimulus. And maybe your body wants to kind of sometimes slow down the gene expression. And so sometimes your body can produce things like miRNAs, which are endogenously, come from within, produced mRNAs, microRNAs, which recognize a mRNA. MicroRNAs recognize a microRNA, form a double-stranded region, and then your body will break it down, kind of so that you can keep making the mRNA so you can respond fast, but maybe you don't need to respond yet. So it's like, hey, let's make the mRNA, but not turn it into protein yet. Let's break down the mRNA with a microRNA, and then when the conditions are right, I won't make the microRNA anymore, and you can make the protein. Or another example is an siRNA, which is a human synthesized RNA that we can insert into cells and use it to silence, that's what the SI stands for, an mRNA of our choosing by being complementary to the mRNA, taking advantage of that evolutionary defense against viruses that I was talking about before. So really, but the main thing I want to talk about is PCR. We can synthesize a single-stranded DNA that is complementary to a region of DNA that we want to amplify, to selectively amplify, make copies. Amplify means make copies of that DNA. So really, it takes advantage of this idea that if we have some single-stranded DNA in solution, and we mix it with a complementary single-stranded DNA, mix it and then cool, it will hybridize and come together to form duplexes again. And so we can go through the cycle of break apart, come together, break apart, come together, break apart, come together. We can go through that cycle again and again and again. And every time it breaks apart, duplicate it. And then we can do that over and over again in order to amplify, make copies of that region. And this is kind of how it works. We have to start out with, and just imagine that this strand here is your whole genome. This is all of the DNA that you have in your body, for instance, or all of the DNA on a chromosome. Runs 3' to 5', 3' to 5'. Let's say that you want to make copies of a region of it, but not all of it. First things first, heat it up to break it apart into 2 strands, 1, 2. And then you can add in a complementary piece of DNA that's complementary for the beginning of the region that you want to amplify on both strands of the DNA. So it's running 5 to 3, and then you can have a little 5 to 3 bit that complements this bit here, and this is running 5 to 3. And you can have a little bit that complements the beginning of the gene here, and you can insert that into the reaction mixture, and then you can cool it slightly, and these will come together, hybridize. Then you can add in a DNA polymerase. Common one is called TAQ, T-A-Q. You can add in a DNA polymerase, and where this bit here, the small complementary piece of DNA will bind at the beginning of your gene, and it will then cool and hybridize. And then you can add in your DNA polymerase, which will then copy in this here, where this small piece of DNA is called a primer. Your primer will bind and anneal when the temperature goes down, hybridize, and then it will, with the polymerase, will then go across and fill in, read across from 5 to 3, read across, and will fill in the gaps against the strand that it's reading from. And it'll do that on strand 1 and strand 2. And then that's a whole cycle. That's one cycle of PCR. So you've got the denaturation, annealing, polymerization. You break apart the 2 strands of the DNA, you anneal or hybridize a complementary small section of the DNA that's complementary to the beginning of your gene of interest. That's called a primer. It will hybridize or anneal, same kind of thing, and bind. And then the polymerase will fill in the gap. This is a cycle. And now you used to have 1, 2 strands. Now you've got 3, 4 strands. You've doubled it. You've doubled the DNA. You started out with 2. You now got 4. And now you've got 4. You can do the same thing again. 1, 2, 3, 4. Except your primer is now going to bind to twice as many. You now got twice as many places for primer to bind, which means now it's going to make 1, 2, 3, 4, 5, 6, 7, 8. Now you got 8. So you got 2, 4, 8, 16. Each time is going through a cycle. Just cycling through, break apart, anneal, duplicate. Break apart, anneal, primer, duplicate. Just keep doing that again and again and again, and you amplify, and you can make your DNA very, very big. You can amplify a lot of it. You don't have to use a primer for this, but you just end up duplicating all of the DNA, and that often isn't useful. And this is used, for instance, for all kinds of things, used in the lab for cloning genes. And if you duplicate a section of a gene, that's known as cloning. That's what cloning is. But also as well, if you were to, in like DNA testing for genealogy and things like that, what you do is that you have several genes which are known, I think there's about 14 genes which are known to be highly variant between different populations and passed on from parent to child. And if you were to just amplify those sections of the DNA, then you amplify them until you get something that's detectable. And if it's present, you get a signal. If it's not present, you don't get a signal. And thus you'll be able to see whether or not it's present or not. And that combination of 14 being present or not is going to tell you whether or not the person, each person is related to each other. Because you know, you may have genes 1, 4, 5, and 6, and another person that might have genes 2, 4, and 9, or something like that. And you can use those combinations to figure out whether or not people are related to each other. Now, the final topic that I wanted to talk about is just about what happens when DNA gets mutated. When you get different changes to DNA that cause damage to the DNA and damage to the genetic material that might need to be repaired. So there's a few different ways that this can happen. It can happen spontaneously, and it could be catalyzed by a bunch of things. And also you can add in various kind of drugs, radiation, that's what chemotherapy is, that cause damage to the DNA, and maybe that's because we want it. But I just want to talk a little bit about the mechanisms that cause this. So first of all, talk about deamination. Deamination is something, it's the removal of the amine groups from the bases. So remember which base has got the amine at the top? 2, 2, 3, 4. Remember which base has got the amine at the top? Oh, this is a pyrimidine, the pyrimidine with the amine group at the top. But this can be, spontaneously can be lost sometimes, and so your cell machinery will need to repair that. This loss can cause a misread, and may cause a mutation, or may cause damage to your cell, may get the wrong thing to get transcribed. This is very, very slow, doesn't happen very much, but you have a lot of it, right? You have a lot of DNA, you have a lot of bases, you have a lot of amine groups. And so there's a lot of, it doesn't happen very much, but if you add up all of that, lots of, not very much, it adds up to being somewhat significant. About 1,000 mutations, for instance, from cytosine to uracil as a result of this deamination. Another one is depurination. And this is where the N-glycosidic bond between the N-glycosidic bond, there's 1 prime, there's the N base, and in the purines, this can get cleaved, or rather over here, my bad. Okay, this can get cleaved, and so you get loss, hydrolysis and cleavage of the N-glycosidic bond. And you can this will get cleaved off and need to be repaired. This is a process, depurination. This happens again quite significantly, but unfortunately your cells can repair it, and it's not so bad to repair because you know that A is going to be paired up with T, and so if you get damage to your A, you can just read backwards and say from the T that's paired up with, and say, well, that must be an A, so I can insert an A in there. There's a few different ways of doing this. For instance, deamination. Cytosine can be mutated to uracil by deamination, or adenine to hypoxanthine, guanine to xanthine by change the amine group, instead changing to a carbonyl because of this chemical reaction, which occurs on a slow rate, but there's a lot of amine groups, a lot of possibilities for it to happen. Depurination, like we're saying, if we have the phosphate base, sugar base, and the N-glycosidic bond can be hydrolyzed. See here, it's not there anymore. It used to be, but it's not there anymore. And so you have you lose the purine. This is known as depurination, and this is now an apurinic residue. A means isn't, so it isn't purinic. It's lost its purine. Another way in which this can, and I don't know whether I don't think I actually mentioned the repair mechanisms, but just to say, one of the common repair mechanisms is that if you have the strand of DNA, and let's say there's something happens and you get depurination here, what your cell machinery will do is actually just make a cut this out, insert a new one in. That's one of the main mechanisms. You just cut it out and put it back in again. I'll put a fresh one back in. That's one of the easiest ways to repair it. There's also other things that can happen. For instance, these are kind of more spontaneous. They can also be induced, for instance, by oxidative damage. Oxidative stress can happen. You can get hydroxylation of guanine, or mitochondrial DNA often gets oxidized. This is because the mitochondria, they produce a lot of ROS, such as superoxide or hydrogen peroxide. And this can cause oxidation of the mitochondrial DNA. And a classic example is hydroxylation of guanine, but other oxidative agents can also cause oxidative damage of DNA outside, like nuclear DNA outside of the mitochondria. You also get chemicals which induce alkylation, for instance, methylation alkylation of guanine. But again, the cell machinery can come in and repair most of these. And a few kind of examples. So this is alkylating agents. These are agents which may cause DNA alkylation, cause DNA damage. Nitrogen mustard will cause alkylation of DNA. Dimethyl sulfate causes alkylation of DNA, so it will react with the things, for instance, amine groups, and cause alkylation. And so thus it's kind of damaged to DNA. It doesn't work properly, and it can cause a mutation. These are chemical agents. These are chemical weapons. You know, World War I, there was a lot of nitrogen mustard. These are chemical weapons which are you know, maybe still unfortunately used today, and they cause DNA damage. But also as well, they could be used as chemotherapeutics, and the same principle is used for some chemotherapeutics as well. To keep along with that theme, for instance, cisplatin and carboplatin are common anti-chemo drugs. They're first-line chemo drugs. Cisplatin isn't used so much anymore. Carboplatin is used instead, but carboplatin is just a prodrug for cisplatin. It's just carboplatin in your body is turned into cisplatin, but it's a lot more gentle on the kidneys, which is why cisplatin has fallen out. But cisplatin is just this platinum, it's cis of these groups, and these groups, because of the coordination with the platinum, you can see it here, coordination with the platinum, cisplatin will pass into the cells, become equated, react with water, swap out that chlorine for this OH2 group, and then this is then highly reactive to DNA and can link together two bases on the same strand next to each other, further apart, or on different, even further apart on different parts of the strand of DNA. And this makes it so the DNA can't unwind because these are alkylated and joined together. DNA can't unwind, the genes can't be read, and then what happens is the cell says, hey, I'm trying to duplicate, I can't, this isn't working properly, and then the cell just dies instead. And so that causes a chemotherapy, because there's a lot of DNA unwinding, a lot of DNA duplicating in cancerous cells, which means that the platinum can, cisplatin or platinum compounds combined can alkylate and then interconnect, and then the DNA can't duplicate properly, and the cell just kills itself instead. And it more effectively targets cancer cells, also targets hair and the stomach, because those cells duplicate a lot, but won't target, for instance, muscle cells, or skin cells get targeted as well a lot, so they get rash, and immune cells, but won't target, for instance, muscle cells so much, because muscle cells don't divide, their DNA isn't exposed, and so they're not as sensitive. Bleomycin is another drug. This is a drug where you can see in gray is in gray is one strand, in green is another strand of our double-stranded DNA, and the drug in red and blue fits in the middle. So this is the center core of the DNA where the bases are, and have that intercalation of the bases joined together, hydrogen bond and bases. Here's the base. What happens is that it intercalates within the bases. So remember, that bond is very strong. It's like a hydrophobic region that intercalates and sticks to the inside of the DNA. And that causes it to the inside of the DNA to get very sticky, so it can't unwind properly. So you get the same effect as you would get here. But also it has these regions that stick out on the outside that generate reactive oxygen species that further damage the DNA. So it's a twofold: intercalating, preventing the DNA from unwinding, and also the generation of ROS, which is causing the damage to the DNA. And then radiation therapy causes a lot of damage as well. So UV light in particular, just on its own without radiation therapy, causes dimerization of pyrimidines, the mechanisms on the next page, but also ionizing radiation, which you can generate, such as X-rays, and also from radiotherapy. That causes generation of free radicals by hydrolysis. It actually just reacts with the water, which is again coming up on the next page. And these free radicals are highly reactive. They break strands of DNA and they open the rings of the DNA, and they're very, very difficult to fix. They're harder to repair than some of the mechanisms mentioned before. And in UV light, this damage to the DNA causes mutations, and these mutations will eventually cause aging and cancer. And for instance, like I was saying, if radiation comes in, it can ionize water and produce these hydroxyl radicals. And these hydroxyl radicals can damage and interact with a DNA. So actually, it's the water in your cells reacts with the radiation and oxidizes it to hydroxyl radicals that then damage your DNA. But it can also directly damage, the radiation can also directly damage the DNA and cause things like breaks, single-strand breaks, double-strand breaks, or loss of bases or joining together of bases as well. It can cause a variety of chemical reactions that damage the DNA. Oh, and for instance, if you were to link together bases of the DNA through ionization reagent, then it causes damage to the DNA. For instance, this kink where the DNA isn't folded properly and can't be read properly as a result. And that covers everything actually to do. We're almost done. I've just got a couple of slides on non-genetic nucleotides. But I just wanted to mention as well, you know, I've talked about this medicinal aspect, these chemotherapeutic drugs. And really what the thing to take away with the PCR and with the medicinal aspects of the drugs is that I told you guys about the aspects of the DNA. I told you guys about the nucleotides, the three parts that make up the nucleotides, how those parts are connected. And then once you have those nucleotides, how they connect into bigger polymers of the DNA, and then how those DNA strands wind together. Told you about those aspects. But then things happen. For instance, you have the breaking apart and the coming together, the complementation, or the chemical reactions that can occur that cause mutations. And really, that's all to do with the chemistry. The chemistry of the nucleotides is being influenced, and maybe the biology influences the chemistry, or maybe the chemistry just comes straight in, influences the chemistry. Environmental factors influence the chemistry. But somewhere the chemistry is being changed. You know, you're getting hydrolysis of bonds. You're getting new covalent bonds coming together, hydrophobic and weakened molecular forces breaking apart. All of these different things, taking advantage of the number of hydrogen bonds to make sure that everything gets matched up correctly, things like that. All of these parts of the chemistry can be modified in order to change the biology, to cause cell death, to cause cell duplication, to cause genetic modifications, replications, mutations, methylation, all these different changes. The chemistry influences the biology as a response to the outside environment around it. The body and biology, life takes advantage of chemistry to adapt to environmental factors, because those environmental factors influence the chemistry influences the biology. Finally, just to finish up, non-genetic nucleotides. For instance, this is ATP, or this is actually just a nucleotide, mono, di, triphosphate. You can have it with adenine, guanine, cytosine, and uracil. You can have it with all of them. Adenine, ATP is the one that you guys are most familiar with, but you've probably heard of GTP as well. If it has a little D in front of it, it means it's deoxy, whether or not it's the RNA or the DNA kind of base. And there's a few different reasons why you have different bases. It's the idea that if you just had AMP as your energy source, well, something may come along that depletes all your ATP, and then you've got no energy. If you were to divide it into 4 different batteries, then something might come along and break and then use each one for different processes. One process may end up sucking up all of your ATP, but you still have GTP, CTP, and TTP, something like that. You have the different ones, and that's going to be that means that you've got redundancy. That means you've got kind of like a fail-safe. Something can use all of this, but you still have all of this for energy. You can divide up the nucleotides into different processes, and then that means that one can't run away and take everything away. Or similarly, for instance, coenzyme A is a nucleotide because you have this. Here's the base, here's the sugar, here's the phosphate, and then you also have this bit attached to it. This is vitamin B1, I believe. Or NAD, here it is. Or actually it's diphosphate, that's what the D stands for, dinucleotide. Or for instance, FAD, same thing. It's a nucleotide with a little bit attached to it. Riboflavin is a B vitamin again. B3 maybe, or B2. I forget, B2 or B3, I believe. One of those is niacin. I think niacin is B3, so maybe riboflavin is B2. I don't know, maybe you can tell me. But they've all got these, they're all based upon nucleotides, have different things acting on them. And actually, there's not that much different between them. The main thing is that they all do similar things. But by having a different structure, you can use them for different processes. So if one process goes haywire, you've still got the other ones working fine on the other processes, and it's not going to deplete those ones. Or they can act as regulatory molecules, such as cyclic AMP, cyclic GMP, different molecules. This diphosphorylated one, PPGPP. So there's various different ones that can act as signaling molecules as well. So not just genetic, also signaling molecules. And I believe that's everything. Thank you very much for your attention.